Meiosis. Have you ever wondered why siblings do not look exactly like their parents or even look so different from each other? The answer lies in the process of meiosis. Meiosis is a process that contributed to genetic variety, and it makes sperm and egg cell, otherwise known as gametes. So meiosis is a division process that takes us from a diploid cell to haploid cells. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes. Meiosis has two phases: Meiosis I and Meiosis II. These divisions have the same processes like mitosis that are prophase, metaphase, anaphase, and telophase. Although meiosis and mitosis have the same processes, there are few major differences. One difference is that mitosis results in 2 diploid daughter cells and meiosis results in 4 haploid cells. But before meiosis can start, the stage of interphase takes place. During interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. Sister chromatids is remained attached at the centromere and condense as the cell enters prophase I of meiosis. So let's start with the very first step, prophase I. This is where chromosomes are going to condense and thicken. They are going to line up with their homologous pairs in a process called synapsis. During this prophase, an event called crossing over can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. In metaphase I, the chromosomes are going to be in the middle of the cell. It's a little different from mitosis because they're still going to be in pairs in the middle of the cell, so it's not a single file line. In anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with telophase I, when chromosomes decondensed and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material and the 2 daughter cells are separated by a cleavage furrow. On to meiosis II. That begins with prophase II, when sister chromatids condense while the spindle starts to form as the nuclear envelope disappear. In metaphase II, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. During anaphase II, sister chromatids separate, and individual chromosomes moved toward the poles. The entire process ends with telophase II. As the chromosomes decondensed and nuclear envelope reforms, cytokinesis occurs and cleavage furrow separates the daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half of its chromosomes from its mother and half from its father. Because men and women produce millions of gametes and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent, nor to siblings.